Every medical test asks the same question: what's happening inside the body?
Bioengineering helps us find answers to this question, whether that means detecting changes in cells, molecules, or tissues.
Specifically, we use a tool called Raman spectroscopy, a technique that uses light to detect molecular changes.
In 1928, physicist C.V. Raman discovered what is now known as the Raman effect. This laid the foundation for Raman spectroscopy.
Here we have a single molecule. Every molecule vibrates in its unique ways. As we can see, the middle part has the vibrations, which are called the molecular fingerprint. Each type of molecule has its own fingerprint. So by looking at it, we can tell molecules apart. When we shine light on the molecule, most of the light bounces back with the same energy. This is called elastic scattering, shown by the green arrows on the diagram. A very small portion of light, however, interacts with the molecule's vibrations and changes energy. This is called inelastic scattering, or the Raman effect, shown by the red arrows on the diagram. By measuring these tiny energy shifts in the scattered light, we can read the molecule's molecular fingerprint, which is the same fingerprint we saw from the vibrations, and learn what kind of molecule it is and how its atoms are arranged.
Now that we understand molecular fingerprints, let's look at a real example. This is a recent study from the National Institute of Health.
Here we can see that each tiny square or pixel represents a specific spot with the x and y axes showing the position across it. At each pixel, a Raman spectrometer measures the molecular fingerprints, which, if you recall, were the vibrations of the molecules. The colors on the map show differences between these fingerprints corresponding to the different types of molecules present. The scale on the side shows the Raman shift, which is the tiny changes in energy of the scattered light that the spectrometer detected. Different molecules and vibrations produce different shifts, which is how the spectrometer distinguishes them.
Looking at the whole map, we can see how molecules are spread across the sample. In bioengineering, this helps scientists monitor drug delivery, study cellular processes, and analyze tissue structure, all using just light.
Fascinated by Raman spectroscopy, I built my own project, which you can see playing in the background. I used Raman mapping to identify what kind of materials were present in the sample, applying the same idea of molecular fingerprints in practice.
By turning light into molecular fingerprints, Raman spectroscopy helps us answer the question that we first began with: what's happening inside our bodies? It helps us understand cellular and molecular changes without any invasive treatments or devices. Beyond biology, Raman spectroscopy also has applications in fields such as material science and environmental science, showing us how a single technology can have impact on different areas of research and innovation.